All right, guys, we are going to take a look at mitosis, and this is all happening inside of your cells, cells of all living things, a lizard, a tree, a flower. Things that grow bigger need to do mitosis, and things that are single-celled need this to make new versions of themselves. Okay, we're going to call those daughter cells. So today when we look at mitosis, we're going to look at the specific phases. And those phases are prophase, metaphase, anaphase, and telophase. Those are the steps that your cells go through to divide the nucleus. So we'll get to actually see this through this animation. And then to finally kind of split our cell, we're going to go through one last stage that will kind of break it apart like we are separating conjoined twins. So if you look inside this cell right now, that's supposed to be DNA. And we call this first stage interphase. And if you guys remember, we actually already did this. Interphase is the super long phase of the cell cycle when your cell is preparing for mitosis. And the way that it does that is copy its DNA, it's going to grow, all to prepare for mitosis, which starts with prophase. So that chromatin, which is our loose DNA, is going to condense into those nice, neat chromosomes. Do you guys remember we talked about chromosomes last class? And we said a chromosome can look like this, like an X, and be replicated, like it copied itself, or it can look like one single worm, and that just means it hasn't been copied yet. So in this example, look at these chromosomes as they sit in the nucleus. That's something we see in prophase. You guys remember the centromere? We said it was that center part of the chromosome where these sister chromatids attach to each other. Each piece of this chromosome is called a sister chromatid. So inside the nucleus, these chromosomes are now visible for the first time, but we gotta get rid of that nucleus, that nuclear membrane, because it's in the way. So in prophase, that nuclear membrane, that nucleus outside part is going to disappear. And we're going to move the chromosomes around now that the nucleus is out of the way. So if you look at those little yellow spindles, they named them microtubules. They're basically little protein ropes that are going to attach to the chromosomes. So we can use these spindles, these ropes, to move the chromosomes where we want them. And in step 2, called metaphase, we're going to line up our chromosomes. Oh, are you kidding me? Okay, sorry, there was an ad. We are going to attach the ropes, the spindles, to the chromosome at the centromere. All right, so they're attached at that middle part. And those ropes, those spindles are going to drag those chromosomes to line up in the middle. So that's how we're going to remember metaphase, middle. Now in the next phase called anaphase, each sister chromatid, each half of that chromosome is going to be pulled apart so that we have one sister chromatid going to one end of the cell and the other sister chromatid going to the other end, so that when we split, each new cell will have a chromosome. So this is anaphase as the spindles pull apart those chromosomes. All right, there's anaphase. That came right after metaphase, which was when we lined up in the middle, and prophase was first. Telophase is last. So now that we've moved those chromosomes to each end of the cell, we can form a new nuclear membrane, a new nucleus around that chromosome DNA kind of stuff that's sitting there. And you guys can see this now. Each new nucleus has been formed around the chromosomes that are going to go back to that spaghetti DNA again. So as we do this, the cell's starting to split. You can see it kind of looks like it's pinching in. That's called cytokinesis. And in cytokinesis, this is going to pinch in the cytoplasm until we split into two separate daughter cells. We call them daughters, not because they're girl cells. That's just our new baby cell. And in a plant, cytokinesis looks a little bit different because of that cell wall. We can't pinch in. So it's going to have a plate that kind of just divides it right down the middle. And we separate into two new daughter plant cells. Not because they're girls, they're just new baby cells. So now each new cell has the same DNA that we started with, right? And that is how we divide the nucleus equally so that our new cells both have all the DNA that we started with.